All right, hello, anatomy people. Here to continue onwards with our discussion on blood. Specifically, today we get into our leukocyte discussion. So, up to this point, you should be familiar with the overall composition of blood, the plasma, the formed elements, the hematocrit. You should be familiar with the idea of erythrocytes, the maturation process that produces the mature erythrocytes, normal functionality, the anatomy, the structure of the cell itself. Um, Also knowing then some of the sort of, I'm not going to say abnormalities, but the natural modifications that can happen with altitude training, hormones that regulate erythrocyte production, maybe some illegal stuff going on with that as well. So once you're familiar with that stuff, now we're able to move onwards with the second category of formed elements that we call the leukocytes. So leukocytes literally translates leuko white site cell. So it's a literal translation from the Greek language, the leukocytes or the white blood cells, white blood cells. So erythrocytes had that one function to transporting gases, oxygen, carbon dioxide. Leukocytes are going to be a lot more dynamic. They're going to be a lot more complex in the types of cells and the roles that they play in the body. So we'll get into those here. So leukocytes will protect the body against microorganisms, toxins, and tumor cells. So basically, leukocytes are components of our immune system. So we're going to deal with immunity specifically later in the semester, but white blood cells are an important integration, an important connection to that chapter. So they help remove dead cells and debris from the body. In contrast to the last sort of lecture, the erythrocytes were not considered complete cells. They don't have a nucleus, they don't have organelles. White blood cells are complete cells. So we do have complete cells. They have all of their organelles. They have a nucleus. So they're regular cells. They're not like the red blood cells. Now, Labster will kind of address this a little bit. And if we were in lab, we would be looking at microscope slides and pictures and sort of ways to identify. But when we identify white blood cells, we look at their size, we look at the shape of the nucleus, we look at the presence or absence of granules and the color that the granules take up. So we're not going to actually get to do this in this format, but you are responsible for the content, the theory behind some of these ideas here, right? So when we look at the normal blood smear, you've seen a picture like this before where, again, we have a normal composition of primarily half plasma, about 45% of the red blood cells, erythrocytes, and then we have a few of these little purple cells. So these purple nucleated cells are white blood cells. And white blood cells will account for less than 1% of the blood's volume. So they're just that little part of that little buffy coat, a very, very small percentage of blood cells in circulation will be white blood cells. So if we're looking at overall hematocrit here on the left, so we have a high amount of erythrocytes, a smaller amount of leukocytes, and then also some platelets here. This is, again, and then the other 45% that would be then the hematocrit. So basically, or I'm sorry, the plasma part of the hematocrit. Now we're just, I mean, it's not to scale here necessarily, but now we're just going to focus on these leukocytes. And we notice that there are a couple of categories. So we're going to define these leukocyte categories. There's going to be 5 different types of leukocytes, right? So for the erythrocytes, there was only 1. For leukocytes, there's going to be 5. Right? And they're going to be divided into 2 categories. something we're going to call granulocytes and agranulocytes. So two categories of leukocytes, granulocytes and agranulocytes. The granulocytes, if you notice, are going to end in the word or the suffix "phil," like neutrophil, eosinophil, basophil. And then the agranulocytes end in the word or suffix "cyte," right? Lymphocyte, monocytes. So what does this mean? Granulocyte, agranulocyte. We have to go back to biology 1306 to make sense of this. So in biology 1306, when you look at the organelles of the cell, I hope you remember something called lysosomes. Lysosomes are basically little organelles that help clean the cell. They help remove damaged parts of the cell. They're organelles that contribute to normal functionality, sort of like a janitorial staff of the cell. Well, some of these white blood cells, some of these leukocytes, will contain these specific organelles, these lysosomes or granules in their cell. We call those granulocytes. Cells with granules, cells with lysosomes. The agranulocytes will not have without, right? A without. So without granules. These are cells that have no granules. So again, I'm going to get in more detail here, but in the overall abundance, there's a little phrase, goes something like, never let monkeys eat bananas. Okay, I think they like bananas, but in terms of anatomy and physiology, we say never let monkeys eat bananas. And that is a little sort of mnemonic that basically lets us know that never neutrophils, let lymphocytes, monkeys, monocytes eat eosinophils, bananas, basophils. So it gives us a way of remembering from the most abundant to the least abundant in percentage of these white blood cells. So neutrophils will account for about 50 to 70% of all white blood cells. Lymphocytes about 25 to 45. Monocytes about 3 to 8. Eosinophils about 2 to 4. And then basophils half to about 1%. So never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils, right? So the overall abundance of the individual leukocyte types here. Hope all this is making sense, yeah? If we go back to the erythrocyte discussion, we should remember this cell. That hemocytoblast, that sort of stem cell, the precursor to all of the formed elements of blood. Well, if hemocytoblast gets stimulated by EPO, they then shift to form red blood cells. If there's different types of stimulations, hemocytoblast can then be converted into one of these 5 types of leukocytes. So it's the same precursor cell, but just gives rise to different types of cells depending on the needs of the body at that particular moment. So we see a very early kind of very different branch off here. So we go from a hemocytoblast to this trajectory then that gives us lymphocytes. If we go this way, we have the myeloid cells in that differentiates in different ways along different sort of pathways to produce then the eosinophils, basophils, neutrophils, and monocytes. So again, you can kind of think that, well, these are a little bit more closely related because they develop along a more similar path. This one is quite distinct. And then the red blood cells are a whole completely different pathway over there. Right? Now for these white blood cells, again, the 5 categories. So never let monkeys eat bananas, right? I mean, here we have our granulocytes, and then the agranulocytes. And just visually, I know they're small, maybe difficult to see, but can you kind of see differences in color? Can we see differences in size? Can we see differences in sort of texture? This is how we're going to distinguish then our different types here. And I'll kind of hop back to this this slide a little bit later again. But let's start with our first, right? So our leukocyte category of the granulocytes. So here we look at our neutrophils, the most numerous of white blood cells, about 50 to 70%. They have the granules, or what we call lysosomes or granules, right? I kind of like to think neutrophils are like the first responders, right? If there's something that's going wrong, we have some sort of infection, boom, the first ones to arrive. The frontline fighters would be our neutrophils. First white blood cells to show up at an infection site. And why are they so abundant? Well, because they are primarily bacterial specialists. And there's a lot of bacteria. Bacteria is like the most common foreign invader that our body is exposed to. So since bacteria is so common, different types of bacteria, different species of bacteria, we have neutrophils to kind of counteract that. So they're there to phagocytize, to sort of engulf, to destroy bacteria. But neutrophils are also effective against fungal infections and antigen antibody complexes, which we'll get into in a later chapter. So first responders really looking to attack bacteria and fungi. So these are granulocytes because they have granules and they look like little spots, right? They look like, if I back up here, okay, neutrophils, I don't know if you can see, it's tiny, but can you see the little spots? Looks like somebody sprinkled pepper on there or something. So they have all the little dark granules to categorize this as a granulocyte. And another weird aspect is that we're going to say they are polymorphonuclear. Polymorphonuclear. So they have a nucleus that has sort of many shapes. So a multiple shaped nucleus, right? Polymorphonuclear. They're going to have between 3 and 6 nuclear lobes in the nucleus. So they don't have just a circular, like a little round nucleus. They have these sort of bilobed nucleus. Why? I don't know. It doesn't really make the cell function more efficiently or less efficiently. It's just somehow the structure of the cell. If you can come up with that, why is it polymorphonuclear? Yeah, maybe I'll throw some bonus points your way. Huh? But they're the first responders. They arrive at the location with this little weird shaped nucleus, all of these little granules. They spend about 10 to 12 hours in circulation, and then they kind of go into the tissue to where the infection actually is. So neutrophils. So first category, right? Next we have the eosinophils, the second category of the granulocytes, right? The phils. And if you notice, I put it in red. Red is a significant color for eosinophils. So let me back up again. So we notice red granules, and we're going to have another polymorphonuclear. There's the polymorphonuclear aspect of the neutrophils. Another polymorphonuclear aspect of the eosinophils. So eosinophils are basically our parasitic infection responders. Right? So if we are unfortunate enough to be infected with some sort of tapeworm, if we ingest some food with tapeworms, with hookworms, pinworms, flukes, some of these large internal intestinal parasites, that triggers the response of the eosinophils. So also involved in allergies in a good way, right? So it's a little bit misleading. Also involved in allergies, I should say they help diminish the allergic response. So they reduce or control inflammatory response by destroying histamine. And I'll talk about histamine in a bit. So they're bilobed. So they're polymorphonuclear, but they're two-lobed, bilobed nucleus with red granules. Right? And 2 to 4% of the white blood cells. So these eosinophils. So red, right? They're going to absorb the red stain, and they have these sort of, I always think it looks like a little, like sunglasses. Another student said they look like a bikini top. But again, bilobe nucleus, or sort of two sort of nuclei connected by that little extension. And we have then the red granules here. So eosinophils. And the idea of, well, they're not very common in circulation. Remember, never let monkeys eat bananas. So they're very low in the list. And it makes sense because in this modern day and age, we don't really deal with too many of these parasites. In our first world countries, it's rare to know somebody that has some of these worm infections, right? So they're not very abundant. I would say in animals, your dogs, your cats are probably more abundant in our evolutionary past. They were very significant, but in modern times, we don't really see too many eosinophils in circulation usually. All right, then basophils, the last of our granulocytes. We look at now our basophils that are going to produce histamine. And if you have allergies, this is what we can blame for allergies. Right now, it's not too bad. Give it a couple of weeks, maybe like by March, where we get all of this wind and dust here in El Paso. Oh, man, their allergies are going to be bad. Maybe yours are not, but mine will be the pollen, things that start triggering allergies. And we can blame allergies then to histamine, which is a vasodilator. And if we take antihistamines, you take things like Claritin, or you take your Benadryl. So those are antihistamines. So they're going to reduce the amount of histamine to try to get that inflammation down. By itself, histamine will increase allergic reactions and increase inflammation. So it basically is like a warning signal. So these little basophils are small. They're not very good fighters of foreign invaders. So they send this alarm chemical, this histamine alarm chemical that causes inflammation, causes this cascade of chaos that tries to get other aspects coming into to help fight this disease. So they have large granules that stain dark to blue or purple. So typically blue, but there was our polymorphonuclear blue. There's a little bit purple, but this is the idea of a basophil. So again, backing up here, backing up. We look then at our, again, red eosinophils, blue to purple basophils, and again, that the neutrophils. So all three categories of leukocytes, all three categories of granulocytic leukocytes, so granulocytes. So with that, let me pause this so the video doesn't get too large. Then I'll continue with the granulocyte discussion and other aspects of white blood cells. So let me stop this one here.